Hi everyone, welcome back to class. This is going to be the last set of notes that will be covered for your first midterm. So your first midterm will cover, the intro stuff, and then acetylcholine, archatocholamines, and serotonin. So make sure you have covered all of that. Remember that Friday's class, Friday the 28th, will be a review class. So I've already posted a Zoom link that you can use. I will be there to answer questions. I'll also go over what the exam is going to look like. So please keep all your questions until then, unless you don't think you'll be able to make it at 3 p.m. on Friday. Again, that will be a live class review. So let's get into serotonin again. In Monday's class, then we talked about serotonin, that it's a monoamine, but not a catecholamine. We talked about its synthesis, its breakdown, and some of its roles, specifically its roles outside the central nervous system. But now we're going to talk about those serotonergic systems and the serotonin receptors. Now, I found this cute meme on the internet today, so I thought I would include it because I thought it was cute and kind of totally relates, right? Actual serotonin in Canto. Oh my god, it's so good. If you guys haven't watched it, oh my god, it's so good. I may have watched it 3 times already. Shh. Moving on. Okay, so here's the serotonergic projections in the brain. So all of these blue ones, you can see that they project or start at a number of different locations, all within the brain stem. And it travels pretty much everywhere, a bit to the cerebellum, but mostly to the entire forebrain. So we get a lot of projections of serotonin. There's a lot of serotonergic cells in the raphe nuclei. So that's very interesting that we'll look at the role of that raphe nuclei in the serotonergic systems. So here we have it. The nuclei is broken down into the caudal raphe nuclei and the rostral raphe nuclei. So in the caudal raphe nuclei, so right here, this projects mainly to the cerebellum and then to the spinal cord. These serotonergic projections help mediate sensory functions and motor functions, as well as those autonomic functions of serotonin. Okay, so serotonin's involved there, but it's not going to be the main function of serotonin that you've probably heard about. So like in that cute meme of like, serotonin makes me happy versus Encanto making you happy, that's not working with that caudal raphe nuclei. That's going to be more the rostral raphe nuclei. So this is, the rostral raphe nuclei is comprised of both the dorsal and medial raphe nuclei. We kind of just group them together. So that's why this picture's it here. Obviously, they're closer together in real life. This is a cartoon. But as you can see, it projects to a much larger area of that central nervous system. It goes to the basal ganglia, to the substantia nigra, that VTA, the limbic system, and just the cortex. We get dorsal projections to the midbrain areas. And that's, again, from the dorsal portion of the rostral raphe nuclei. And then cerebellar projections, so right here, this tends to go more to the deep cerebellar nuclei, as well as the cerebellar cortex itself. So it's going to different places of the cerebellum than the projections from the rostral raphe nuclei. Now, because this rostral raphe nuclei is involved in more of the, I guess we'd call it cognition almost, or more of the brain areas, we're going to really focus on the rostral raphe nuclei system. So what happens if we were to lesion or cut these serotonergic projections? Well, we can do this in one way, would be to send in some neurotoxins that then block the serotonin, and we can then see, well, what functions is serotonin doing? Okay, so it's an experimental way. So parachloramphetamines and MDMA both are neurotoxic effects for serotonergic pathways. So we can do this at either very high doses or do a micro injection. So in rats, we do a stereotaxic surgery, implant that cannula, and then do a micro injection into that rostral raphe nuclei and see what happens. Or we can use this drug, 5,7-DHT. This is a blood-brain barrier impermeable selective toxin. So since it's impermeable, then what we can do is, again, do a micro injection straight into the brain. You can't do really an oral dose, right? But it can induce a robust serotonergic injury. So what we see is that when we provide one of these neurotoxins, either type, then we see deficits in food intake. So we talked about the role of serotonin in eating before. We see reproductive behavior deficits, pain sensitivity, anxiety, learning, memory, and motor function deficits. So, what is happening with serotonin? What does all of this mean? And what we see is that if we focus on just that dorsal portion of the raphe nuclei, then we can see what's happening with the activity of that dorsal raphe nuclei at different times of the day. So recording the activity of these cells there, then we see that when they're awake, they have a steady firing pattern. And then as they go more into sleep, then that activity decreases. Okay? So frequency increases with motor activity, but a sudden sensory stimulus can cause an abrupt sensation. So say we're here and it's a little slower and then something suddenly happens, then we'll get like a peak of activity. So it seems like the dorsal raphe nuclei is involved in activity of some sort, in motor activity or something along those lines. So there must be some sort of motor effect that these serotonergic neurons are having. And what we've kind of assessed is that these serotonergic neurons facilitate motor control, and they're proposed to suppress sensory processing to facilitate repetitive tasks. Okay, so serotonergic firing patterns are stopped during sensory processing. So presentation of a novel stimuli will disrupt the tonic firing of the DNR. So that DNR is the dorsal raphe nuclei. Okay. So the effects of serotonin during these motor functions contrast with the firing of the locus coeruleus norepinephrine neurons, okay, which are important in performing repetitive tasks and are active on presentation of stimuli. So the ability of serotonin to suppress sensory processing during a repetitive task can be linked to the ability to zone out when performing these repetitive tasks. Okay, so during repetitive tasks, there's kind of a balance between the norepinephrine and the serotonin in these neurons. Okay, so the locus coeruleus is there to be like, hey, we're doing the repetitive task, but then the dorsal raphe nuclei and its serotonergic neurons are involved in the ability to zone out during those repetitive tasks. And you only get a spike of activity during these repetitive tasks when there is like a novel stimuli. Okay? So, serotonin also has broad behavioral effects. It's not just happiness, though that is the one big thing that serotonin is attributed to. Okay. So serotonin is colloquially thought to contribute to happiness, hence that meme at the beginning of class. And many antidepressants do target serotonin reuptake and actually helps improve mood. So there is a correlation with serotonin and happiness, but that's not all it does. So serotonin binding capacity has actually been suggested to correlate with a tendency towards spirituality. So, how does serotonin relate to spirituality? There's this compound right here. It's a numbered compound that was developed by Wayleth Airst, totally. And it targeted the serotonin 1A receptor. Okay, so this is the spirituality receptor. And it was a serotonin 1A receptor agonist, so it eggs it on. And it was used as a PET ligand to identify serotonin receptors in the human brain. What they saw was that a 2003 study from Swedish group correlated that this receptor binding capacity with components of the Temperate and Character Inventory, which is a self-reporting questionnaire that includes questions about spirituality, and they did this in male volunteers. And they found a significant correlation between spiritual acceptance and that binding capacity. Okay. And in particular, the authors proposed that the serotonergic system may provide a biological basis for these spiritual beliefs, and why, spirituality is global, worldwide. Many different cultures have some sort of spirituality. And the observations of several fold differences in the serotonin 1A receptor binding across individuals in a healthy population could actually explain variations between people in their different levels of spirituality. So why some people are more spiritual than others, or more susceptible to spirituality. Okay? Is that there's actually different like polymorphisms or different folded variations of this serotonin 1A receptor that may be related to degree of spirituality. It's so cool. So not only is serotonin associated with happiness, but also spirituality. So I mentioned the serotonin 1A receptor with spirituality, okay? But you're like, Erin, we haven't talked about receptors yet. Silly me. Here's the list, okay? There's so many receptors. There is a mix of G-coupled protein receptors and ligand-gated, okay? This is a very large family of receptors. There's 14 different receptors that have been identified, and we group them into 7 different groups, okay? So serotonin 1, 2, 3, 4, 5, 6, 7. Those are the families. And then they can have other ones. So 1A, 1B, 1D, 1E, 1F. Great. Serotonin 2A, 2B, 2C. And then 5 is the only other one with a thing. You'll notice that serotonin 3 receptors are the only ligand-gated channels. You also might notice that there's no 1C. Interesting, right? Well, it turns out there used to be serotonin 1C receptor, but then more research was looked, and we actually discovered that it's not a serotonin 1 family, it's actually serotonin 2. So serotonin 2C used to be serotonin 1C. but it was reclassified because of how it works. So notice that serotonin 1 receptors are all GI, so they inhibit, whereas serotonin 2 are Q or I. They can be either, but they work on PLC and increasing calcium, whereas the serotonin 1A receptors act on CAMP and GERK. So because serotonin 2C receptor does not act on CAMP or GERK, then it was declassified as a 2. I hope that makes sense. Okay, let's go ahead and stop this video here, and we'll start the next video going over some of the specifics of each of these receptors.